All right, so good morning, everyone. We're going to get started. Welcome to the parameters of biological validation, which is the kickoff for a workshop that's going on, the biology of biosignatures detection. My name is Bethany Ellman. I'm the Associate Director of the Keck Institute for Space Studies here at Caltech, which is a think and do tank with the idea of revolutionizing how we do space exploration, both the science of how we explore our universe, as well as enabling breakthrough technologies. We look for ideas that are far horizon, but are going to be large impact. And of course, one of the most profound questions is the question of, are we alone in the universe in terms of the search for life? So I'm very excited to welcome the folks from this workshop, which is led by Scott Pearl of JPL, Charles Cockle of University of Edinburgh, Woody Fisher here at Caltech. But importantly, for every single time that we have one of these workshops, we have a short course, both to bring everyone in the workshop up to the same page, but also to share what we're doing with the Caltech and JPL community and the community of people here in Pasadena and the LA area who might be interested in the community of people listening online who follow what we do in our workshops. So welcome, everyone. Hope to learn about biology here, and I will turn it over to Scott for the introduction of what you will hear about today.
Awesome, thank you, Bethany. Excuse my voice, it's returning after field work. But thank you to everyone for being here. Thank you to the speakers, as well as everyone that's involved with KISS, as well as the visitors that are here today. And this is something pretty special in terms of how long this has been in the works, both in terms of the actual effort to bring this on board, but a lot of the actual discussions that we actually formed this from have been long-standing, and so years ago to actually have these conversations, and now we're all sitting here because of those conversations, and so it's pretty important to note that. And so when it comes down to what we actually think about in terms of life detection, it tends to be a single point that really doesn't actually take into account biology. And so when we're going through biological validation, what the point of this actual series here is today, as well as the entire week's effort, how do you validate something where we have a sample size of 1? And that's something that is always going to be a problem until we're able to actually make solutions to solve those problems. And so the way that we actually do this is through these actual lecture series, but also understanding what we would need to validate signs of life outside of our own world. And so I will kick it off now in terms of our first speaker. And so we have Karen Lloyd. And so Karen is coming to us from the University of Tennessee, soon to be USC, and she will have our first talk. And so, Karen, when you're ready.
I'm moving to USC in August of 2024, so that's going to be fun. So I'll be in this area. I wanted to start with a definition because when Scott told me about this workshop, I did not actually know what an agnostic biosignature was, so I was thinking maybe I wasn't the only one. So what it is basically just evidence of life that's independent of any of the specifics of Earth life. Like, it doesn't have to be DNA. It's something universal and agnostic. It's independent of any preconceived notions that we have of the specific examples of life. So the problem with finding an agnostic biosignature is that you want it to be not specific to Earth life. You want it to be all of life everywhere, but you also want to avoid being so general that you don't actually distinguish between what's living and non-living. Like, threading that needle is actually quite difficult. There's a lot of self-replicating chemical systems or crystal lattices that really have a lot of the attributes that we normally would associate as being living, but of course they're not. So here's some examples, because I hope this this lecture is not too general for the audience. Well, whatever. You can pull out your laptops if it is. But these are some things that I think of as not agnostic indicators. These are the things that we use all the time to detect life in the subsurface of Earth that may or may not be relevant for life elsewhere. DNA is my favorite. I use it all the time. RNA, proteins that have 22-ish amino acids, lipids, metabolites, specific sets of metabolic reactions, chlorophyll, other photopigments. These are things that we use here on Earth. But there's also some indicators that are probably signatures of life that well, I guess I want to point out, sorry, before I want to point out some, we sort of know that these are a frame of reference that may or may not be universal. But I think that there's some other frames of reference that we don't even think about. They're just implicit, like temperature and pressure. We sort of have this 122 degree upper temperature limit of life, and we probably have a pressure limit for life in our brains. But it's important to not see that as an absolute limitation. For instance, Tommy Gold had some out there ideas, but in his less out there 1992 PNAS paper, he posited that life could have a silica-silica bond instead of carbon-carbon bonds at higher temperature, for instance. Another one that I think about a lot, and I want you guys to think about because I think it's important, is time. We have this framework in our brains that drives me nuts of how long an individual can actually be alive. We kind of cut things off at about 100 years. But a lot of what we're doing in marine sediments in the deep ocean show that there's just not enough power for life to replicate on any kind of good time scale. So I think we need to knock that out of our brains because we could get something that looks like a biosignature on another planet or planetoid, and it just looks like it's sitting there and doing nothing, and we may chuck it out the window as a biosignature when actually that thing is just waiting on the next millennium or hundreds of millions of years. I mean, there might be some crazy timescales happening somewhere else. So I think we have to keep that in mind. And I don't have any evidence to back this up, but we should look at the shorter time scale too. I mean, I say often, life can't compete with an explosion. Why do I think that? There could be very fast life too. Again, zero data to back that up, none whatsoever. So these are things that I think are agnostic, but are too general. These are things that we think about as being signatures for things that life has to have. Life has to catalyze or speed up the destruction of potential energy, the use of potential energy. We know it has to do that. Lots of chemical reactions do that too. Catalysts for chemical reactions do that too. We have to create information systems to organize ultrastructures. That also happens in an eddy in a stream, for instance. We see ultrastructure that maintains a stable state in non-equilibrium conditions for longer periods of time, and we all know that an eddy in a stream or a whirlpool is not life. So we have to find a way to distinguish it. And self-replicating with autocatalysis, with DNA and cells. And obviously this can be done chemically as well. So these are not new problems. I looked through the literature to find some background for this. And I found this paper from 1962 by Herbert Simon. And I want to read that first paragraph because it's like, I don't know if it's depressing that we're exactly where we were in 1962, but he got it just right. He says, a number of proposals have been advanced in recent years for the development of general systems theory, which extracting from properties peculiar to physical, biological, or social systems would be applicable to all of them. We might well feel that while the goal is laudable, systems of such diverse kinds could hardly be expected to have any non-trivial properties in common. Metaphor and analogy can be helpful, or they can be misleading. All depends on whether the similarities the metaphor captures are significant or insignificant. So here I'm here to talk about analogies to the life in our current subsurface on Earth, but that may or may not be relevant. What he concludes in this paper, if you read the rest of it, is that one feature of life is that it's a hierarchy of subpopulations. And that has an important step in allowing evolution to happen, that you need like semi-metastable states in parts. And so looking for a hierarchy of subpopulations may be something that goes into an agnostic indicator. I also wanted to bring in the view of Ilya Prigogine and Isabel Stengers from 1984, moving into the modern era. Basically, I'm summarizing what they say. In far from equilibrium systems, stable structures like eddies in a stream can be maintained by constantly creating energy gradients. So this is a feature that we know that life has, is that we are constantly organizing and passing information around to continue creating energy gradients. We destroy the energy gradients to get energy, but then we make more. So on the net, we end up creating even more energy gradients than we potentially destroy. And that is a feature of life. Sort of my last sort of from the literature moving well into the modern era with someone who is both alive and still a scientist is Joe Volino at the Marine Biological Laboratory. And I guess I won't read through this point of his abstract, but he has an idea that I really appreciate, which is that the point of life is to to maximize total entropic production from an ecosystem or from a system by extending the time scale over which entropy production occurs. in so doing, you can actually create more entropy than you would if you just burned your thing today, if that makes sense. So I think this is an intriguing feature as like something we can put our finger on and say like, this is a thing that life strives to do, is just spread out entropic production in order to maximize it. So non-equilibrium thermodynamics gives us these features that are agnostic and specific, but it's hard to connect to things we actually measure. So this is what we have to do. I'm coming here to this workshop and I'm like, okay, what are we going to measure? We're going to look for patterns in hierarchical, the Simons thing, information-rich systems that maintain themselves far from equilibrium and maximize entropic production by extending it over longer timescales. How do you measure that with a gas chromatograph? Like I've said like a very general thing here. What do you put on an instrument that flies? So it's hard to connect those two things. But I think that the trade-off is between agnostic features, not blinded by the specifics of life, specific features that can distinguish between life and non-life, and then that third axis is measurable stuff. And we need to find the center of all those things. So because this is a general lecture, I'll just tell you what we know about subsurface life. Again, I really hope I hope you learned something new. If this is really boring, just stop me. The goal of my talk, as I have been told, my goal is to highlight the nutrients and potential metabolic processes of subsurface microbiology. So first of all, what are the nutrients that life needs? I divide these up into two categories. One is that molecules that can react and microbes can conserve energy from them. And then molecules that contribute to the building material for life. I think it's really important to separate out these two categories of things, because when we talk about nutrients to a person who works in photic systems, they're almost always referring to the building material version, because their energy source is not confusing, it's the sun, and that basically drives everything. But when we talk about life in the subsurface, which may become increasingly relevant for life outside of Earth, our energy source is just as intriguing a question, what's driving it. And those two things do not have to be the same biomolecules at all. Things that are energy nutrients are pairs of compounds or elements that are either good at giving up an electron or taking an electron, so redox reactions. Things, just off the top of my head, that fall into this, almost everything in the transition metals. If you think it's just iron, like toss that out. It's really, almost anything has been, even gold has been used as an electron acceptor for microbial reactions. So lots and lots of parts of the periodic table. I specified for, I didn't put methane in there, I should have, for carbon, just because carbon's so general. But in general, it's any reaction that is exergonic, that yields energy, but slow. And slow is, of course, a relative term, depends on what you're talking about, the time scale. It has to be able to be sped up by life, and then it can be used by life. So the easiest pair is hydrogen and CO2. This is all you need to make, plus the building blocks. But energetically, that's all you need to make life go. You can do acetogenesis or methanogenesis. And so that's one reason why it's so intriguing. So other things you can do to get energy are single compounds that can be split into a more oxidized molecule and a less oxidized molecule. And the best studied version of that is when we do it with organic molecules. So this is fermentation. This is something that obviously we know tons and tons about. But this is something that also applies to subsurface life or life that happens outside of photosynthetic material, because organic molecules can be synthesized abiotically. So this is something that could be sort of primordial. Also, you can have stable inorganic molecules of intermediate oxidation state that split into a more oxidized and less oxidized version. So disproportionation. And sulfur, elemental sulfur is an example of something that can do this. And the third category would be two molecules of different oxidation states combining to make a single molecule. And this is comproportionation. The best studied example that I know of this is called animox, and it's where you take electrons from ammonium and put them onto the compound nitrite, and then you end up with N2 gas from this. But more recently, Jan Amund has shown that in his group that you can do this with sulfide and sulfate as well. So generally speaking, this stuff has a huge energy range available. We know that it goes down to minus 10 kilojoules per mole, which is the bleeding edge. As far as we know, that's the lower boundary for how life can get by. Could be wrong. Up to minus 3,000 kilojoules per mole is what we get at standard state from breathing oxygen, which, you know, you can do all the things that we do, tennis and laughing and things like that, heating up a room when you walk into it. So it's just a gargantuan amount of energy. The other side is almost always pretty low energy. These comproportionation and disproportionation reactions are not going to yield a big whale. You need something more powerful for that. But I want to point out that we often talk about energy with this, like, we stick in our brains to the Gibbs free energy, which is all about equilibrium, and equilibrium is death, right? So we got to get out of that and think about not just energy driving. Sorry, I'm talking over myself. We need to not just say low energy things are less likely to support life and high energy things are better for supporting life, because we know that it's not about the total energy, it's about the delivery rate of that energy. So you can be just fine on low energy as long as you are super high turnover. And we see this with production of food. I mean, one reason why we have all the leftover alcohol that we can make all these nice drinks with is because Saccharomyces cerevisiae is like next to death on every reaction run that they do, so they have to make a whole lot of this stuff. So you can actually compensate for low energy with power. So ironically, sometimes these low energy processes could have a bigger effect on the biomolecule composition of the community simply because they have to turn over a bunch of times and leave a bigger impression on the chemical fingerprint. So that is an important point that is sometimes lost, I think. So squeezing all that into that side and going to the building blocks, schnapps, we all know that, right? You got to have those to make life happen on Earth. I don't know if that is a specific Earth thing or if that is a general thing that you need for life. No idea. Perhaps you guys know more than I do. You also need a lot of metals. And the good thing about looking in the subsurface for this is that actually these things are more abundant usually in the subsurface than on the surface of planetoids. There's a whole lot of fighting that happens up in the photic zone of Earth for almost everything in this list, but in the subsurface it's plentiful. It's a really great place to live for that. It's a little tougher on the energy. Okay, so these are the things that are necessary. So where do we find them on Earth? Where do they come from on Earth? So if we start to look on other places for sources of these nutrients, how will they be like where they're found on Earth? So I'll divide this into two categories. You can tell from the tiny writing that I'm about to put up a lot of text here. This is what happens when you write it with jet lag at the last minute because you wrote the wrong talk. Either stuff that falls in from the surface or stuff that comes from the subsurface, top down or bottom up. So stuff that falls in from the surface is plant matter, anything from primary or secondary production from photosynthetic life, and I include in this ancient reservoirs of dead stuff, so petroleum, coal, that kind of stuff. That stuff was originally photosynthetic. So even though in our modern era we do subsurface work to dredge it up, it was originally from the surface. This type of nutrients are plentiful because the sun is such a powerful energy source. The building blocks here can be limiting, but otherwise you get a ton of biomass. So example ecosystems where we find this, my favorite one is marine sediments. It's a massive ecosystem on Earth. The total number of microbes that are present in the deep subsurface of marine sediments is equivalent to the total number of microbes in all of the world's oceans. So this is a huge population. And also subsurface aquifers that have heavy recharge with a frequent rewetting from groundwater, and coal, petroleum, methane deposits, things like this. Okay, so going to the other category, things that come from the subsurface that don't depend on light from the sun, which somehow seem more relevant to, at least when we're talking about our own solar system, because I feel like if there were a lot of trees on Europa, we would know by now, I think. I think we can rule that out. So I'm thinking subsurface is going to be very important for our solar system. Serpentinization is everybody's favorite reaction for a good reason. It's not just because like some people got some grants to study it and they're like wayward. This is a seriously powerful reaction, and it may be the key to the origin of life on Earth and life elsewhere in the universe. This is a simple water-rock reaction. It creates heat, it creates fractures, it dewaters minerals, leaving a space behind. It inflates the total volume, so it basically causes minerals to burble up to the surface where we can get them. But most importantly, it creates hydrogen, and then later downstream reactions can create carbon dioxide, methane, carbon monoxide, and this is an iron, oxidized iron, and this is very, very prevalent in ultramafic rocks that are silica rich, that are not silica rich, like pyroxene and peridotite. So this is very powerful reaction. And then there's other hot water reactions, water rock reactions that happen in mafic systems as well. And there's also degassing of mantle and downgoing slab. These are sources of hydrogen CO2 that can happen even independently of serpentinization. We can talk about whether a downgoing slab is relevant, because as far as I know, there's not a lot of plate tectonics that happens outside of Earth in our solar system. But there's at least probably some mantle, maybe on other planets or planetoids. I don't know. Is there? Well, I don't know if we have enough time to. We can talk in the Q&A. Somebody can tell me if there's mantle. But our mantle is a good source for some of these gases that can support life. And we've found ecosystems that basically are living solely off the CO2 that comes from mantle degassing. Radiolysis of water produces hydrogen and sometimes CO2. This is something really good that came out of the Cold War. When we started having a lot of nuclear waste to store, people noticed that there was a lot of gas being produced in their nuclear waste. But this process happens even with background radiation from the radioactive decay of Earth. So this is a constant and steady source of nutrients that are getting made in the subsurface. There can be abiotic thermal synthesis of organic carbon. That used to be pretty controversial, but I think it's not so much anymore. And we have evidence that there are chemolithoautotrophy-based ecosystems that basically are happening independently of the sun, but we don't have ancient reservoirs of biomass from them. So I don't think they ever were like the Cretaceous, where there were just tons and tons of biomass that can be used for fuel later, but they are there. So example ecosystems are hydrothermal vents, natural springs, deep ancient aquifers that don't get a lot of recharge. These are the places that we find them. The good news about these types of ecosystems is that they tend to produce fluids that pop out to the surface. And so this is something I do a lot of my work, is using those as windows into the deep subsurface biosphere and trying to distinguish from more recent groundwater sources. Okay, so given all these things, all this background about what nutrients organisms need, how can we detect the subsurface life agnostically? So we can come up with new measurements to make, and I think that that's a really good idea, but I don't have that in my brain. That's something that I'll hear from other people, hopefully. What I have in my brain is how to find innovative ways to measure data types that are already possible to measure, and working with existing data streams to find out something more fundamental about life detection. So going into this Simon analogy can be helpful or it can be misleading. All I've got is analogy. So how much time do I have, Charles?
You still got
15 minutes. Okay, sorry, I shouldn't have talked so fast. So I want to talk about analogy. I want to talk about how in my work, in the work that my colleagues and I do, of course it's never on my own, we use data that are easily measured and commonly measured, but put it together in ways that can help tell us about biosignatures of the subsurface without making a new measurement. So the analogies that I'm going to talk about that will hopefully not be misleading are in sort of two categories. One is co-correlation networks. Actually, I don't know whether to call them that or correlation networks. But we use DNA, RNA, and other metabolites. And I'll tell you about two examples where we looked at Costa Rica, hot springs across Costa Rica, to detect subsurface life, and then also to put together metabolic pathways without actual organisms. And then there are machine learning techniques which we're doing, but are unpublished, so I'm not going to put it in this part of the talk. That's basically, you can think of like machine learning as correlation networks on steroids. It's ways to find patterns in things that are more complicated than what you can see with your own eyes and easily detect with your own brain. And then you should be very skeptical of the results that you get and make sure they make sense, because you get spurious results all the time. So my analogy is, this is basically the whole country of Costa Rica, just to give you a sense of the scale. And every dot on here is a different, I want to say hot springs because that's the right idea, but some of these were actually quite cold. We don't just go for the super hot stuff, and we're using it as deep indicators of deep life, or at least subsurface life. So when we take samples, we're not sampling from a pool that's been sitting at the surface for a long time. We're going directly to the source, and in some cases, like shoving a pipe down into the source. So we get microbes that have never seen the light of day, ever. And this is a massive collaboration. There's even more people than I've listed up here from my group. It's been Kate Fullerton, Joy Bongiorno, and TJ Rogers who've done most of the work. But really, the analysis that I'm going to show, laser pointer's gone, but I'll just use this, is from the bearded dude right there, Donato Giovanelli. This is pretty much his analysis. Oh, there it is. So one thing we struggle we have is that we are collecting, this is collecting a gas sample. We're collecting these samples right at the edges of rivers. There's a lot of potential input of the surface world into what we're sampling. You can see these iron oxidizers, Gallianella, are making these twisted stalks, and there's so much of it that it's just leaving this massive imprint on the rivers. If you ever wonder, I mean, I think we're all settled on where BIFs came from in our brains, but here you can see like modern future BIF formation by these iron oxidizers. It's never been mysterious to me about how they produce such huge deposits just because they make so much. Here's our data stream. It's a big bunch of nothing. You can't, you get data. This is identities of all the microbes in all of our springs. Here's the different springs lined up here. Here's the names of all the microbes. We can categorize them, figure out who they are. The size of the circle tells you how relatively abundant their DNA sequence is. And we line them up with temperature, because you can imagine that like the temperature is going to really drive the ecosystem. And that's true. We see that there's these guys are hotter, these guys are cooler. But how do you actually, the question that's relevant for us right now is how do you use this normal data stream to figure out what's actually a life detection signature for subsurface life versus stuff that fell in from the surrounding areas, the jungles. So you have to reduce the complexity of the problem because I can't see patterns in that. I can't just look at that. And we divided microbes into groups that correlate in abundance to see how they relate to the geochemistry. So what Donato did is that he made this giant co-correlation network. And if you're not used to looking at these, each little dot you can imagine as a microbe. It's a DNA sequence, but just see it as a microbe because we're using 16S RNA genes here, which are kind of like name tags. 2 dots that are close together are co-correlating in abundance across these springs. So 2 little orange dots sitting next to each other are things that if one was there, the other one was there. And if they both, if one decreased in abundance at the next spring, the other one decreased in abundance at the next spring. So from this, he was able to identify groups of co-correlating organisms. And then to look, actually he did use some machine learning to figure out which geochemical features they correlated with. And they correlate with organic carbon. This one does. This one correlates with organic carbon as well. And this one correlates inversely with inorganic carbon. And so by doing this, there's a whole study behind this, but just to show you what we did, laboriously through every single different group, we were able to see ones that are potentially the main primary producers. So this group of organisms are the ones when the plate is deep. These are close to the volcanoes. The progression of subduction is sort of how we're dividing things, dividing these springs up. And they're using iron and sulfur for chemosynthesis, we think. And these are heterotrophs that go along with them. And then we can identify these other groups up here that are primary producers in the other part. So this is out near the coastline in early subduction when the slab is shallow and there's a lot of sulfate around. And then these guys, this was sort of, you'd think this is maybe a negative result, but for me, this is the most important part for our current discussion, is that we were able to see organisms that did not correlate with any geochemical parameter. And so we think these ones are just washed in from soils. And when because it's Earth and we know a lot about the microbes, we can see what their names are and say, okay, those are normal soil microbes anyway. So that's what we would expect to see. But we also got some normal soil microbes that are in these deep subsurface groups too. So in that way, we were able to sort of, if correlation is a biosignature, then we can see a biosignature of what's happening in deep samples that is correlating with the mantle gases relative to what's up at the surface. And I know sample size, you need a lot of different samples for this. And usually if you're sampling on another planetoid, you only get one planet. But if you've got a rover sitting up there, you can potentially do lots and lots of measurements over time. So we found that the 16S showed us the subsurface versus surrounding soils. So the second example that we did is, so this is now taking a metagenome. So this is all the DNA present in these samples. And we presume a lot of times about, we say, okay, this gene, we can say that this gene is here, and we know that in a cultured organism, it follows along this pathway and works with these other genes. But we wanted to say, if we don't know anything about how these genes interact with each other a priori, how do we figure out which genes are interacting in the same pathway as each other? And so we took just the total reads of these genes. This is kind of technical, but if you're a metagenomic person, we did this read-based before we ever assembled it. And we annotated the reads and then made a co-correlation network here of all the genes that are related to carbon cycling in these systems. So each dot here would be like a different gene. And what we found is that we got these 3 groups out of here, and it just so happens that the genes that group together are genes that belong to a product that operates in a pathway for carbon fixation together. And so fully sort of agnostically to what those genes do, we didn't impose that on the system. We didn't say, we know that you're a carbon fixation gene and you should be working with this other gene. We just said, I don't know, I think you're a carbon fixation gene. What are you doing? And we saw that it emerged that some of them correlated with each other. And so that implies that they're functioning in a pathway. It's not proof that they are, but it at least leads you to that point without imposing your preconceived notions on the system. And then we found that this middle group, which is the Wood-Lungdahl pathway, which was very prevalent in our organisms, did have its best correlation with this inorganic carbon if you add all these up together. So implying that this community, the chemosynthesis of this community, was dependent upon this carbon coming out of the mantle in the slab. So this is just summarizing sort of what we found, that the distribution of microbes showed us an ability to distinguish surface from subsurface, and we were able to see which ones potentially work together in a metabolic pathway. So back to this question. How do you look for patterns in hierarchical information-rich systems that are extending entropy? How do you do this with the gas chromatograph? Well, the way you do it is that you make a ton of measurements and then figure out how things correlate with something else that you want to know. And I know that's a really general suggestion, but I think I'm making it general on purpose so that we can, because I don't want to say, I think it's useless to come into a workshop like this and say, my measurement's the best, we should do my measurement, which is not always the best for the science. But my approach is best, and we should apply it to whatever you measure. I don't know, that's kind of a joke, but that's the notion, is that take whatever you got in terms of measurements and just be smart about how you analyze it, and then you can discover these signatures. And I want to thank all am I too early? But that's cool. You guys probably like to talk, so that's good. I have received funding over the years from all these fantastic sources, and I cannot get my lab in one place. I don't know if y'all have this problem. So this is like some part of my lab, plus some parts of other labs. This is just my most recent picture, and I'll be happy to take questions.